Introduction. As Dennis said, my name is Jennifer Sneed. I'm a research biologist here in Fort Pierce at the Smithsonian Marine Station. And what I want to talk to you guys about today is the importance of bacteria and ecological interactions. They're tiny, we can't see them, we don't think about them most of the time, but they're very important. This picture here, just to kind of explain it, is actually the picture of a bryozoan larvae. And this ring of cells are all bacterial cells within that bryozoan larvae. And you'll see this picture again later and have a better idea of what that's about. But I just want to start off by talking a little bit about marine bacteria in general. One of the reasons they're so important is just that they're so abundant. So in 1 milliliter of seawater, there are 1 million bacterial cells. So that means in the vial this big, I'm holding up right here, there would be 1.5 million bacterial cells. That's a lot of bacteria. Okay? On the surfaces underwater, so if you go out to any marine environment and look at anything basically that's underwater, whether it be natural or unnatural, so boat hulls or algae or rocks or whatever, they're all covered in marine bacteria. And on average, there's about 10 million bacterial cells per centimeter squared on those surfaces. So a centimeter squared, I measured earlier today, is about the size of my thumbnail. So depending on how big your hands are, you can think it's about the size of your thumbnail. 10 million bacterial cells. That's a lot of bacteria. So not only do we have a lot of bacteria in the ocean, but they make a lot of really important compounds. And so this is a figure from 2006 that said there are 1,008 known compounds to come from marine bacteria that are what we call bioactive. That means they have some kind of influence on other organisms. So they're compounds that bacteria produce that influence other organisms. So we have a lot of bacteria, and they have a lot of really important and interesting compounds. I'm going to talk today mostly about the role of bacteria in ecology because I'm an ecologist and that's what I'm interested in. But I just wanted to start off touching on a little bit of why they're really important to people as well. And so they're important because they can be producers of drugs. So we're looking a lot for pharmaceutical compounds now from organisms in the oceans. If any of you saw Dr. Amy Wright's talk, I think it was sometime in the past year, she talked some about these compounds that they're looking for here at Harbor Branch. In our lab, we look at compounds from the ecological perspective, but then we also, if we discover new compounds, we send them off to be tested for potential pharmaceutical agents. And right now, there's a couple of compounds that are being tested for pharmaceutical activity that come from marine bacteria. And this is one of them. This is bryostatin 1. This compound was originally isolated from this bryozoan. So that's why it's called a bryostatin, because it was isolated from a bryozoan. But it turns out this compound is produced by those bacteria that I showed you on that first slide. So these bacteria that live within the bryozoan are the true producers of the compound, of this compound bryostatin 1. And this compound is currently in phase 1 and phase 2 medical trials for anti-cancer and anti-Alzheimer's. It's looking particularly promising as an anti-Alzheimer's drug. There's another one called Selenosporamide A. This comes from this bacteria, Selenospora tropica. So this is actually showing how it grows on an agar plate, so that wouldn't be one cell, but a huge colony of bacterial cells. But this compound is also in anti-cancer trials currently. So these guys are really important in terms of the compounds they produce that could be important for future drugs. But they have a lot of ecological importance, and like I said, that's the area that I'm more interested in. So I want to talk a little bit today about, in general, the kinds of roles that bacteria can play in ecological interactions. And then towards the end, I'm going to focus in on some of the work that I've been doing here at the Marine Station. So when we think about bacteria in the marine environment, there's basically three different kinds. We can talk about these kinds that are planktonic. And planktonic means in the water column. So when you're talking about anything, be it fish or microalgae or bacteria, anything that lives out in the open ocean in the water column is called planktonic. So planktonic bacteria are really important in nutrient cycling. They can produce some interesting compounds for defense and for competition, so to help them defend themselves from being eaten or compete within their environment. Benthic bacteria are a little probably more studied. They produce some more interesting compounds. These are things that are associated with the soil and with the surfaces underwater. So they're either actually incorporated into the soil on the bottom of the ocean, or they're on the surfaces, like on the surfaces of algae or rocks or whatever surfaces might be under the water. And basically, like I said earlier, all marine surfaces are covered with bacterial biofilms unless they specifically have some mechanism for keeping off a biofilm. A marine biofilm will form on any surface you put under the water. And they live in a very competitive environment. So because there's so many of them there, I told you there are 10 million bacterial cells on the surface as the size of my thumbnail. They have to compete for space and nutrients and all those sorts of things. So it becomes a very competitive environment, which leads them to produce a lot of compounds that help them to live and compete in that environment. And so these kind of bacteria produce a lot of different secondary metabolites. They can produce antimicrobial compounds. So those are compounds that would actually inhibit the growth of other bacteria. They can produce anti-fouling compounds. Those are compounds that keep things from settling on their surface. This is an area that's really interesting to shipping companies, the Navy, things like that, because boat hulls are constantly being fouled by algae and barnacles and all sorts of things, and they're always looking for different kinds of compounds that they can coat onto the bottom of a boat hull that will keep things from settling on it. And so that would be a kind of anti-fouling compound, something that keeps things from settling on the surface. And they can also produce settlement cues, so they can produce compounds that cue different organisms to settle on a surface. And then we have the endosymbiotic bacteria. These are ones that actually live inside other organisms. And these, really interestingly, are often the true producers of compounds that were previously attributed to their hosts. So just like I told you in the beginning that the bryostatins were thought to be produced by a bryozoan, it turns out they're actually being produced by the bacteria inside them. And we're finding out more and more that that's the case as we start to develop more technologies and be able to look at these microbes more closely. We're finding out that they're very active in producing compounds that were previously thought to be produced by their host. And so they can actually mediate interactions between organisms in the environment in a number of different ways. So if you think about this organism A that you have living under the water, and then you have this whole big pool of bacteria out there in the seawater, so this big, vast bacterial community that's made up of all kinds of different bacteria. So first of all, this arrow in the middle, the color of this arrow is going to indicate that these are compounds. I'm talking about compounds produced by this organism. So organism A could produce nutrients or chemical attractants that attract certain bacteria to their surface. And so those bacteria come and settle on the surface. They can also produce chemical deterrents like antibiotics that keep off other different bacteria. And so they can selectively regulate which bacteria live on their surface. So when you look at different organisms in the water, you often see that different organisms have different bacterial communities living on them. But then those bacteria that live on the surface of the organism can play a lot of roles in the ecology of that organism. So the bacteria themselves then can start producing compounds that have a lot of different effects. So they could produce antibiotic compounds, so that would block some of these bacteria from coming to the organism. And an example is the elkhorn coral. It has this bacteria in its mucus. It's a bacteria in the genus Marinobacter. And that bacteria actually produces compounds that inhibit the growth of this bacteria, Serratia marcescens, which causes the whitepox disease. So this bacteria that it has in its mucus is defending it from disease. It could also deter feeding. And this was a suggestion from Laura Diedrich that I make this into a little Pac-Man. So they can deter feeding, amazingly enough. So let's hope it works. It was a last-minute change. So again, we're back to this bryozoan that I was showing you earlier. It has the bacteria in it, the Endobugula certula, that produce those bryostatins. And the bryostatins are in anti-cancer trials, but they were discovered because they have an ecological role. So they actually, the bryostatins inhibit feeding by fish on this bryozoan. And so this happens a lot. This is one of the reasons I said earlier that when we find a compound in our lab, we send it off for drug testing, because often those compounds that have activity in ecological systems are also the ones that are potentially active in anti-cancer, anti-HIV, anti-tumor, all those kinds of things. But this is another cool example that this bryozoan actually has bacteria that live inside of it that keep it from being eaten by fish. The bacteria that could produce compounds that inhibit settlement by organism B. So for example, the sea lettuce, it has this bacteria on its surface called Pseudoalteromonas tunicata. And these bacteria keep our bryozoan friend, again, he's showing up a lot in this talk, they keep this bryozoan from settling on the surface of the algae. And so you can think that's really important to an organism that needs to photosynthesize. It doesn't want to have things growing on its surface and shading it, basically. It needs the light. So it's important for this sea lettuce to have these bacteria that keep this bryozoan from living on its surface. And they can actually induce settlement. For example, coral larvae. And this is where I'm going to jump now into a lot of the research that I've been doing for the last couple years in the Smithsonian, looking at the effects of bacteria on coral larval settlement. That's going to be the next half of my talk. This is the larva from a staghorn coral. This is a staghorn coral. This is his larva. And just when he's settled and metamorphosed, this is what he looks like. But I want to back up a minute and just make sure everybody is on the same page and understands what corals are and why they're important. So hard corals are important because they're reef builders. So they actually build a calcium carbonate skeleton, and they build the reef, and they allow the wide diversity that lives on a reef to survive there. And part of the reason they can do that is because they come in a lot of different shapes. So you can see there's two different branching corals shown here. They have a lot of different places that fish could hide, all kinds of different environments. And then there's also these big boulder corals. So there's a lot of different shapes. And there are very different sizes. You can see this boulder coral is very large, while this little golf ball coral is actually about the size of my fist. And to some people, it might look like a rock sitting out there on the reef, but it's not. It's actually an animal, and it's a colony of individuals. So if you look a little closer, you can start to see all the little circles. Each one of those is an individual polyp. And if you look even closer, that's what they look like up close. So they look kind of like the polyps in there look kind of like sea anemones, and they're related to sea anemones, so that's why they look similar. But they actually have that hard skeleton surrounding those polyps. So then I always throw up this horrible statement or the horrible quote in my talks about this stuff. Coral reefs are in danger, and I'm sure most of you know that already. This was a quote from the Nature Conservancy that says 70% of the world's reefs will be destroyed by the year 2050. That's a pretty somber prediction. However, there are a lot of people working very hard on coral restoration and coral science to try to understand how we can reverse this trend, and hopefully we'll make some progress in that direction. And one of the ways that we can do that, if we want to think about how we go from this degraded reef where there's nothing living back to a healthy reef where you have lots of biodiversity, you have to have corals, right? You have to basically plant corals if we want corals to grow there. So that's being done through some kinds of restoration efforts, but it's also important that recruitment happens naturally. And so that's kind of what I'm studying, is the natural process of recruitment to a reef. So how do corals come in, settle, attach, and grow on a reef? So to understand that, you have to understand a little bit about the life cycle of a coral. And basically, there are two different life cycles. There are broadcast spawners and there are brooders. And I'm going to take you through each one of these separately. So the broadcast spawners, they release gametes into the water. The gametes float up to the surface where external fertilization happens. Once external fertilization happens, the zygote develops into a planula larvae, and eventually that larvae will swim to the bottom, it will attach, and it will grow into a coral. So to show you what that looks like, I'm going to take you through a series of pictures. This first one is showing you Zach Foltz. This was one night when we were out looking for coral spawning. So when we work with the spawners, we have to go out after sunset. It's usually 2 to 3 hours after sunset when they spawn. So we go out and either dive or snorkel and start looking around at the corals. And what Zach's looking for here is to see whether the corals are going to spawn. And then if we see that they're going to spawn, then we can start to work with them. And so this is what it looks like when they spawn. This is a video of coral spawning. You can kind of start to see there's little pink bundles in the mouths of all these polyps. So you'll see in just a minute they're going to release them all. So do you see that flush of pink bundles that just got released? Those are the gamete bundles that the corals are releasing. Those gamete bundles have sperm and egg inside them. They'll float up to the surface, and that's where they'll break apart. Fertilization will happen, and then the coral will grow from there. And so this is happening all over the reef. When we're out there at night looking for these, if we have a good spawning year, we had a pretty good spawn. I was down there about 3 weeks ago. We had a pretty good spawn. And you're out there basically in what's like a snowstorm of gamete bundles everywhere, because their corals all over the reef are releasing their gametes at the same time, and they have to release them at the same time so that cross-fertilization can happen. So when that's about to happen, we go ahead and throw a net over the coral to try to catch all those gamete bundles. And you can see, I hope, that at the end of this net we have a cup, and the gamete bundles float up, they get trapped in that cup, and then we can take them back to the lab and start to work with them. So we take them back to the lab, and we go ahead and separate the sperm from the eggs, and then we do cross-fertilizations. We don't really want to fertilize the eggs and sperm from the same colony together. They're much more likely to be healthy and competent if we cross-fertilize between different colonies. So this is Dr. Nicole Fogarty. Many of you might know her. She was a postdoc at the Smithsonian, and she's here cross-fertilizing different coral colonies with each other. So this usually ends up happening, I don't know, midnight, 1 in the morning. We're fertilizing, getting the corals ready. And then we put them in these buckets in the flow-through lab, and we let them sit there and grow, basically. And so while they're sitting there, they are growing from this into a fully developed larvae that we can use in experiments. And depending on the coral species that we're working with, that can take between about 2 to 7 days to get to competency so that we can use it in an experiment. Brooders, on the other hand, the other kind of corals, they actually take sperm in and have internal fertilization. So all that fertilization process happens within the coral, and then they release a fully developed swimming planula larvae. Makes it much easier to work with. So these guys, like the mustard hill coral, it's a brooder. When we work with these guys, we work mostly in the Keys. We bring them into the lab, actually, and so we can set them up in this flow-through seawater system. We put the coral into, actually, a mixing bowl, very scientific. But this mixing bowl has this nice little water slide here. So we can put a flowing water here, and the water will run down that handle. And we put that handle inside a beaker that has mesh on the bottom. And so we can set that up at night. We go off, we have dinner, go down to Key West for a minute, come back. Next morning, we have a beaker full of larvae. So you can see that's a little bit more blown up. You can see each one of those little brown dots is a fully developed larvae. Much easier than working with the spawners, but not quite as fun. And this is what one looks like magnified. But the result of both of those kind of reproductive processes is that you have a swimming larvae that's then going to swim down to the bottom and find a place to attach. It will metamorphose, and then it will grow into a coral colony. And that's the part that I'm particularly interested in. How does this little swimmer that's swimming around in the dish turn into that? And how does it pick where it's going to do that more specifically? So I'm interested in how does that larvae choose a place to attach and metamorphose. So we know there are some different kinds of things that coral larvae might be attracted to. Sometimes they're attracted to coral rubble or other biofilm surfaces in the water. Sometimes they're attracted to conspecifics, so that means they would be attracted to settle near something of their own species, which makes kind of sense because you know if mom lives over there, it's probably a good neighborhood. I can go live there and I'll be healthy and happy. But often they're known to settle on this crustose coral and algae. So if you ever look at rocks in a fish tank, in a seawater fish tank or under the water, and they look like they have a pink paint on them, that's actually an algae that's growing on their surface. It's called the crustose coral and algae. And I abbreviate it CCA, and so I probably will refer to it mostly as CCA from now on. So if you hear me say CCA, that's what I'm talking about, that pink film that's growing on the rocks. So coral larvae amazingly can respond to a lot of different stimuli. They can respond to color. They can respond to light. They actually swim away from the light when they're ready to move down to the bottom. So that's how they know which direction is down, or one of the ways they know. They can respond to sound. They've been shown to actually swim to a reef or to the sounds produced by a reef, which is really interesting. Once they get down to the bottom, they start to probe around and look for a place to attach. And so once they've done that, they can respond to texture on the surface, and they can also respond to chemicals. And I bet you can guess which one I'm most interested in. It's the chemicals. So I'm really interested in the chemicals associated with the crustose coral and algae that the larvae are using to find a place to settle, attach, and metamorphose. And so Raphael Ritson-Williams, who many of you probably know, and Dr. Paul, who's here tonight, they did some work before I came here looking at different species of crustose coral and algae. And it turns out that larvae prefer some species of CCA over others. So they're not all created equally. And this work was done with larvae from the elkhorn coral here. But how do they know? So how does a larvae know this is one species of CCA and this is another species of CCA? And there's a lot of different ways that they might be figuring that out. But I'm a microbiologist, so I thought maybe it's the bacteria. Maybe these CCA have different bacteria living on their surfaces, and the larvae are able to distinguish between those. So one of the things I did when I started here was to actually look at those CCAs and see if they had different bacterial communities. And I know this graph is very sciencey. Don't worry about all the different scientific names. I'm going to step you through the graph. But basically, I looked at the same three species of crustose coral and algae that they had looked at before. So Titanoderma prototypum, Hydrolithon borgesonii, and Paragoniolithon solubili. And when they did these assays, they found that Hydrolithon borgesonii was preferred more than the other two, followed by Titanoderma. And then the larvae almost always picked something other than Paragoniolithon solubili. They don't really like this guy very much. So basically, I just looked at the bacterial communities that were growing on their surfaces. And these pie charts can just show you, if you just look at the colors, that they're not the same. They don't have the same proportions of different kinds of bacteria on their surfaces. So each color is a different kind of bacteria. And you can see that, for instance, this has a very small blue piece of pie, and this one has a very large blue piece of pie. So they're just, the point is that they're not the same. They do have different bacterial communities, which means that they could be a way for bacteria to recognize, or for larvae to recognize the surface, if they actually have distinct bacterial communities. A couple of things that I found interesting in this data were that there's this huge blue piece of pie in these actinomycetes, and actinomycetes are known to be producers of a lot of compounds, a lot of active compounds. And the other two have actinomycetes also, but just not as high a percentage. And there's a couple other bacteria that were found only on these two preferred settlement substrata, and a couple that are found only on the non-preferred. So it could be that these bacteria that are found only on the non-preferred are actually repelling the larvae from that crustose coral and algae. But overall, what this told me is, yes, these CCAs have different bacteria, so it's possible that the larvae are able to figure out which one is which based on the bacterial that are there. So then I was forced to go to some really horrible places to do a lot of work. I did most of this work at the Caribou Cay Marine Field Station down in Belize, and at the Mote Marine Lab in Summerlin Key, Florida. And it's tough. It's really tough. But even though these are beautiful places, we do a lot of work down there, and some of you have been along with me on these trips, and you know that that's the case. And so I'm going to show you some of the data from that work. So one of the first things I wanted to do was find out if the larvae needed bacteria to settle. So would they settle on something that didn't have bacteria on it? So what I did was take these tiles, put them out on the reef. I told you already, you put something underwater, bacteria grow on it, right? So I put those out there for 3 weeks. I let the bacterial communities form on their surfaces, brought them back in, treated half of them with antibiotics to knock back those bacteria, and left the others the way they were, and then looked at how the coral larvae responded. And this involved a lot of counting. So we count the larvae when we're putting them in the treatments. So we have our tiles in a little dish, and then we count 50 larvae per dish, and then we let them sit for 24 hours, and then we count them again. Let's have somebody here in the audience helping me count. We count these again to see how many settled and how many are still swimming. So here you can see these have that kind of flower shape, and these are still swimming around. And I did this with both brooders and spawners, 5 different coral species. I apologize for the scientific names. I couldn't figure out how to get them off the graph before I came here today. But this is the mustard hill coral. This is the golf ball coral. So for both of these, what you see on this axis, and I'm going to have a couple of graphs like this, but this is just the percent settlement. So this is how many settled. In this case, the blue bars are that natural biofilm, and the red bars are antibiotic treated, so where I removed the bacteria with antibiotics. And you can see both of these settle when they have bacteria present, but when you remove the bacteria, the settlement goes way down. The spawners aren't all quite the same. So the two branching corals here, this is the elkhorn coral and the staghorn coral, they didn't really seem to care if there were bacteria or not there. I think they really wanted another cue besides just the bacteria. But this brain coral here, it also settled at higher amounts on the tiles that had bacteria than those that didn't. So again, it says bacteria might be important. They might be responding to bacteria. So is it just any bacteria? Can I put any bacteria on a tile and they'll settle in response to that? Or are there specific bacteria that they respond to? So for this, I isolated a bunch of bacteria from the surface of different CCA species, and then I actually coated these ceramic discs with the bacteria. So each ceramic disc had one individual bacterial strain on it, and looked at how the bacteria responded to all these isolated bacteria. And so I did this with 23 species. You can see them here across the bottom. And what stands out right away is that, yes, one of these strains induced settlement. So none of those other bacteria induced settlement. The larvae did not respond to them at all. They weren't interested, nothing. But this one, PS MBA5, induced settlement. So then I wanted to find out what it was. Turns out it's in this genus, Pseudoalteromonas. And this is just the name I gave it. It was part of this original name. So Pseudoalteromonas spa PS5, that's my guy. He's the one that induces settlement. What's really interesting about this is that I isolated this compound in Florida from a bacteria in Florida, or I isolated this bacteria in Florida, and it induces settlement of this Caribbean coral, Porites asteroides, which is the mustard hill coral. This bacteria is very closely related to one that was found in Australia that did basically the same thing. It induced settlement in these two Australian Pacific coral species. So it's really interesting that we both found, completely separately on opposite sides of the world, a very similar, closely related bacteria that has the same, does the same thing. So the next thing I wanted to find out is if I could find out if it was a chemical cue that was being produced by the bacterium that induced the settlement. So I grew the bacteria on agar plates, and then I scraped it off with a razor blade. I extracted the compounds from the bacteria, and I tested just the compounds. So now we have no bacteria, but just the chemical compounds that the bacteria produce. And I looked to see if they induce settlement as well. So we added the compounds to sterile seawater. So these 6-well plates, each well has sterile seawater in it. And we added just the compounds, nothing else. And then we added larvae. These are actually larvae from the elkhorn coral. And then we went back and looked 24 hours later to see if they induced the larvae to settle. And in fact, they did. It's a little hard to see in this picture, but these are all attached just to the plastic in that 6-well plate. So with just that chemical signal alone, no other stimulus, they're settling in response to this compound. And they don't settle if you give them just seawater or just seawater and the solvent that we use to add this extract in. They don't respond at all. But with this extract, they settle and metamorphose. And so we did some work to figure out what compound in there was causing them to settle. And it turns out it's this compound tetrabromoperol. And so just to take you through the process again, this is the swimming larva here. And then this is, after we give them the compound, they settle and attach. And then we actually went back 4 weeks later and looked at those larvae to see if they were growing and if they were healthy. And this is what they look like. And so basically, this is a little baby elkhorn coral right here. You can see it's laid down this calcium carbonate skeleton. And it's got its little polyp coming out here. And it's actually taken in zooxanthellae, which are its symbionts. It has a symbiotic algae that it doesn't have when it's a baby. So that's why it's white and clear here. But as it develops, it will take in its symbionts, and that's why it's brown here. And so you can see this nice little polyp with its zooxanthellae, and actually 2 little buds coming off. So there's one here and one little one there you can barely see kind of around the corner. So it's happy, it's healthy, it's growing, it's got zooxanthellae. The compound is not harming it at all. It's just inducing it to settle. So I've done this experiment now with 4 different species of Caribbean corals. So the mustard hill coral, the elkhorn coral, staghorn coral, and the boulder star coral. And this compound induces settlement in every coral that I've tested it with so far. So some future research questions. I want to know if this compound induces other species. I know that it induces the 4 that I've tested it with so far. Are there any other species? Is it ubiquitous? Does it induce the settlement in all Caribbean corals? Also, I want to know how common these bacteria that induce settlement are in the environment, and also how common that compound is, that tetrabromoperol. Because we did all this work in the lab, but now we need to find out, is this really what's acting in the environment? Are these bacteria there in large enough numbers? Is the compound there in the right concentration to actually be a signal to coral larvae when they're looking for a place to attach? And I'm also really interested in how climate change and other environmental stressors will affect these important marine bacteria. So I think there's a lot of research being done on the effects of climate change and stressors on corals and on other macroorganisms, on polar bears and their territories, and all of that is very important. But I just want to stress that let's not forget the bacteria. The bacteria are doing so much behind the scenes, and I think we need to do a lot more research into how these kinds of things are going to affect bacterial communities. We might cause increases in disease-causing bacteria or decreases in bacteria that the corals need to find their home. So we need to keep that in mind as we're looking at how these different environmental stressors are going to affect marine ecosystems. And with that, I'd like to thank a lot of people. I couldn't possibly list all the people that have helped with this work, that coral spawning work and counting all the larvae. It's very labor intensive, required a lot of students. But I'll try to point out at least the people that are here today. Dr. Valerie Paul, Raphael, he's not here, but I feel like I should point him out as well. Lindsay Spears is here tonight. She did a lot of help with larval experiments down in the Keys. A number of other people. I've had a few interns that have worked on this project from IRSC. So Heather Granger, Eric Weil, and Jay Hauk, who's here tonight. I need to thank them for all their help. I couldn't do it without help, obviously. It's a lot of work. The Smithsonian Marine Station, the Caribbean Coral Reef Ecosystems Program, Moat Marine Lab down in Summerlin Key, and also their Protect Our Reefs grant program, because they funded a lot of this research. And with that, I would be happy to answer any questions.